Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is April 25th, 2024. It's a special day for us at UTSA because it's the day of our Annual Neuroscience Symposium. And this year's symposium's title is Epigenetics and Neurodevelopment, and we have 5 experts on epigenetics and neurodevelopment in various proportions to speak to us today. And we listened to 5 great talks today, which the podcast audience won't get to share, but which were great. And I think we're going to have a discussion because we just finished listening to this stuff. And it's all really fresh in our minds. So let's start by having everybody introduce yourself. I'll start with you, Paul.
I'm Paul Ramirez. I'm a soon-to-be graduate of UT Health San Antonio in the Best Frost Laboratory. And my work revolves around retrotransposons and epigenetics in Alzheimer's disease.
Okay, I'm Melanie Carless, an associate professor in the Neuroscience Developmental and Regenerative Biology department at UTSA. My work focuses on epigenetics of complex diseases, including those related to neurodevelopment and psychiatric disorders and neurodegenerative disorders.
I'm He David Xie from Virginia Tech, a professor in the Department of Biological Science and Pathobiology. My lab is a computational biology and epigenomics lab. We primarily using mouse models and cell culture to study how brain cells get specified and how they gain their function.
Great. Hello, listeners. My name is Ladd-Acosta, and I'm an associate professor in the Department of Epidemiology at the Johns Hopkins Bloomberg School of Public Health. And my research mostly focuses on genetic and epigenetic epidemiology with a particular focus on autism and developmental disabilities.
And I am Alexei Soshnev. I am here at UTSA. I'm a chromatin biologist, and I primarily work in mutations that impact chromatin architecture in cancer, but more recently I've been branching out into neurodevelopmental disorders.
I'd like to get us started just by trying to pin down exactly what epigenetics means and what's included in that field. And so maybe that sounds crazy simple, but usually these things turn out to be more complicated than they sound. And so I'm going to start by just giving the most naive possible interpretation, which I know can be corrected. Alexei Soshnev has corrected it a couple of times in the past. I'll try to be better this time. And
third time's a charm.
I think I would just try to define by saying the entire set of cellular processes that are responsible for controlling gene expression in a differential way, so that one cell expresses one and one another. And I know that's very broad because it includes a lot of very classical things. Maybe you might even try to include the enzymatic machinery of transcription into that definition, and you probably wouldn't want necessarily to include that in epigenetics. But correct me, what is epigenetics exactly?
I think a narrow definition of epigenetics is transmission of information in non-DNA sequence-dependent ways. So in a narrow definition, it is about intergenerational transmission. And which is why what we do here for the most part is not exactly epigenetics, it's more chromatin biology. But in a broad definition, you're absolutely right. It's regulation of DNA activity by stuff that's around it.
So what are that collection of mechanisms? So today mostly we heard about 2 kinds of things. One is methylation of DNA, and the other was histone modifications. And but in the course of talking about it, lots of other things came up. Things like enhancers and transcription factors. So, and do we know all the mechanisms? Are there undiscovered mechanisms that will someday be considered part of epigenetics? I think particularly about the three-dimensional structure of the chromatin.
I don't know. I guess when I think back on, when I was in graduate school, for example, and thinking about what we understood and what we knew about epigenetics then, it was nothing compared to what we know now. So I think we'd be naive to think that we found it all and we know all of the mechanisms. I think there's more out there to be discovered. I like to break them up into like DNA-based mechanisms, RNA-based mechanisms, and kind of protein-based mechanisms. It gives me three different classes to look at, but within each of those, there's so many different ways, in which things can be controlled.
Could we try breaking those down a little bit? Because
okay, I can take the DNA mechanisms. We can pass it around and see how we go. So for me, the DNA mechanisms, again, as you mentioned, we spoke a lot today about DNA methylation. It's definitely one of the most studied mechanisms. This is where you just basically get like a methyl group attached to a cytosine molecule. Most of the time in mammals, it happens at what we call these CPG sites, which just means a cytosine and a guanine nucleotide next to each other. But it also happens at other sites, particularly in the brain. We get methylation, still at cytosines, but not necessarily when they're next to a guanine, but potentially when they're next to other nucleotides as well. And then we have kind of these processes that allow for demethylation that kind of take you through this oxidative process where you get these intermediates that also have different regulatory roles as well.
And I asked this question during the talk, and I'll ask it again. The methylation alters transcription by interacting with the transcription factors? Or
it helps to condense. It interacts with, these protein factors that can kind of help condense the chromatin, making, that gene inaccessible to transcription factors. And even just the act of having that methylation there again sort of reduces the ability for the transcription factors to bind. So typically what we see if we get this methylation at the promoter regions of genes, is we see this inactivation of gene expression. So increased methylation corresponds to, decreased gene expression. But then there's also sites that are not in those promoter regions that have different functions, potentially, involved in alternative splicing. Some of them will actually increase gene expression. And then I think as well, we just don't know what a lot of those things do.
So to get a handle on sort of how much we know and how much we don't know about this, I'm just thinking, what's the total proportion of these methylation sites that are at promoters, where we have some kind of notion of what they do, and how many of them are at places that we just don't know what they do? We can fess up, right? We don't have to.
Yeah, I think the numbers I'm not sure of.
Yeah.
I can give some numbers.
I know.
I think it's around 20. I think if I recall correctly, there's 28 million CPG sites in the genome, something like that for human.
Yeah.
Mouse slightly less than that. But the finding on the genome is quite clear now. Like or is promoter, or enhancer keep growing the knowledge, but because our histone modification chipseq and is able to identify the histone modification marks and able to annotate genome much better. Now I think it's much more clear what other regulatory elements are in the genome. So I think the whole picture become more and more clear. But from epigenetic term, I like what Alex mentioned there, but I want to emphasize again. So one is that you don't need to change DNA sequence. You change the gene, how gene work, right? Don't change. And the other is intergeneration, like one cell pass to the other cell. And if you call it epigenetic, you need to have a mechanism to replicate the mother cell. For example, DNA methylation, we are talking about, then we have a DNA methylation enzyme. When the DNA replicate, the enzyme DMT will copy the mother strand DNA to the daughter strand DNA. That is really important.
Including all the methylations.
That is, and then the other thing is that it's dynamic. It can change inside the cell, right? Even the cell don't change in the DNA, but it can change in the epigenome. So there's a couple features that define the epigenetic term. And then we get.
I would echo that real quick because we talk about CPG methylation all the time because CPG is symmetrical. If you think about the DNA, it's a CPG on Watson strand, it's a CPG on the reverse strand. And so a maintenance methyltransferase when DNA gets resynthesized recognizes a hemimethylated pattern and can reinstall at the CPG site. If it's just a C, it doesn't know if it was methylated there or not. So you need to have this symmetrical structure. And in the brain, where DNA replication is not really a thing, because in a neuron you don't have to do that very often,
It's terminal
the
differentiation.
non-canonical C methylations come in. Now you're talking about CHH, CAA, all these other funny other types of methylation, which are, honestly, we don't really understand them, but they're fascinating.
Maybe I can add a bit of my perspectives too on all these. So, it's interesting because I think you're right. Over time, what we mean by epigenetics has changed. And even if we go back to the 1940s, right, when Conrad Waddington was recognized as sort of the father of epigenetics, right, that's before we even knew that DNA, right, was the heritable material. But he was a developmental biologist and actually recognized this concept of something's inherited, right, and the concept of genes. And then there must be something in between those genes and the phenotype, or what we observe, that kind of controls, right, that coming into being. And so I think, even back then, it was this really broad kind of idea that there was some cellular material, or lots of material that kind of sat between the genes and the outcome, right, or the phenotype that controlled those and brought it into being. Then I feel like for a long time, I learned back in probably the 80s to 2000s, it narrowed. To be very specific, I would say definitions of, well, it's methylation and it's histone kind of modifications. And I felt like, again, it's broadened a bit now, to be more inclusive in recognizing there are other kinds of information and components of cells that are really important, again, in kind of bringing the phenotype into being. But I also learned, I think the key things, though, that or criteria it should meet it has to be mitotically heritable, right, which was already brought up. So when a cell divides, it helps the cell remember what its lineage is and what kind of cell it should be. They can be reversible, and they are anything other than the actual DNA sequence itself.
So we normally think in neurons that they are not dividing, but during development, that neuron precursors are dividing.
Mm-hmm.
And so a neuron lineage that's going to give rise to some particular cell type can change over and accumulate changes over the course of development in order to start from the first approximation to a cortical pyramidal cell, and then finally ending up with the complete cortical pyramidal cell at the end, as the cells that are destined to do that go through divisions. Is that not?
I'd love to hear, actually, from you all. I'm not sort of trained in neurobiology, but, my understanding is really neurodevelopment occurs not just in utero, but really even through mid-20s, right? And so, what does that look like, and how does the cell development change over that amount of time? It's a pretty broad time.
So,
And then there's adult neurogenesis, which is debatable, so I don't want to get into that.
Yes, and that too.
Jenny's not here, so.
So we think that the methylation can survive these cell divisions. How about histone modifications?
I can answer that. There's some fascinating papers. It started with Danny Reinberg's lab at NYU. And they tracked down, during the same replication fork when the DNA is being doubled, what happens to the nucleosomes at the replication fork? Because obviously if you are making more DNA, DNA polymerase has to go through, you have to reassemble the nucleosomes. You can't just replicate keeping the nucleosome there. And what they found was pretty fascinating, that the histones that contain activating marks are associated with open chromatin get pulled off of DNA and then they go elsewhere. They don't stick around. But the repressive marks, namely K9 methylation, K27 methylation, stick around the replication fork. They don't go too far. And so as the replication fork progresses, histones that contain repressive marks get recycled at the same site locally. They don't know the mechanism. There are some options for that. But that might be one of the mechanisms to retain repressed domains. And then, of course, there was some fantastic work from Johns Hopkins about the asymmetrical histone retention during germ cell division. Drosophila, Xin Chen lab has been working on that many years. Lots of interesting stuff.
So how about offering an example of a mechanism we don't know too much about that we know exists? Perhaps like long-range interactions between DNA or weird things that have to do with the structure of the heterochromatin?
Sure. I'll take a stab at one. Maybe this is an example you're looking for, but one of the functions and roles, I think, of epigenetics, an easy one to conceptualize and that we understand is what we call X chromosome dosage compensation, right? So females have 2 copies of an X chromosome. Males have one copy of an X and 1 copy of a Y. And so because females have this double dose, they need to silence, right, one of the X chromosomes. And the way that occurs is through epigenetic mechanisms. So there is kind of random coding of the X chromosome with methylation. It involves methylation. It involves recruitment of a long non-coding RNA exist, and also chromatin, right, heterocompaction. So I think on multiple levels of what we would consider epigenetic information, they're all working together to really kind of clamp down, right, and silence that extra X chromosome so that females don't have double dose kinds of issues of genes being expressed.
And super quickly add to that, that in humans, we chose to select one X to silence it. In other organisms, which have the same problem, X chromosome can get double activated in males. Flies activate their X twofold to match the female dose. And other organisms actually figure out a way to half express, half express. So they actually regulate both female X's, but drop the expression level about 50% in both of them. So there's many ways to solve the same problem, and evolution has come up with multiple options for that. It's fascinating.
So the phenomena that we imagine will ultimately be understood by going down this line of thinking are things like cell type definition, what determines what cell types there are and how many there are and where they are, morphogenesis, maybe the craziest, tough problem, but which really comes from the cell type issue. If you start making the right type of cells, they arrange themselves into a morphologically identifiable thing. In the cortex, you know, think about that kind of stuff. Also, developmental defects and developmental errors and mistakes that you can understand. Learning and memory, which we don't really know everything about learning and memory, but we can imagine that memories require some change in expression of genes in order to have permanence, to not just disappear the next time we fall asleep or something.
And there's definitely a lot of genes whose methylation levels, are associated with those processes of learning and memory as well.
And there are diseases, like in the nervous system, we think about psychiatric diseases that have very low inheritability, but some, and that I sort of imagine are probably because of some kind of genetic.
Well, and often is the case is you actually see sort of this high heritability for these psychiatric disorders, but when you go in and you look at what are all the sequence changes that are associated with that disease, And for some of them, I mean, that hundreds have been identified, but the amount that they contribute to that heritability is like often only maybe 2% or up to 10%. And so, we have this idea of what's the missing heritability, which, can be how those changes interact with each other, but then also these epigenetic factors play a role as well in controlling that. And so there's sort of this much larger picture than just thinking about like what's in the genes. It goes far beyond.
And I think different environments, right? There's evidence that environments may impact epigenetic changes, right? Sort of outside of genes. So that might be important in contributing to.
Like environmental toxins in food.
Exactly.
So there's I guess it's the corporate motivation, collective motivation, I should say, collective motivation for us trying to study epigenetics and understands it. It goes beyond just our curiosity about how cells work and develop and faces like serious social issues and diseases. I wonder, because this seems like such a huge question, maybe we're not going to really understand the stuff I did about morphogenesis or what makes a cortex. The cortex, super quickly, maybe there's some shortcuts. So understanding the molecular mechanisms that are underlying might offer the possibility of shortcut treatments or cures for something. What's your personal strategy? Would you rather like settle into the long haul, try to solve the big question, or would you like to get some useful benefit out of it?
I mean, I do both.
Yeah, that's what
I don't want to put all my eggs in one basket. I think there's still so much to be discovered, and that, kind of those questions can drive you forever. Like, you can do this for as long as you want, trying to find the answers to those questions. But as you said, I feel like we get into this business because we want to help people. We want to be able to treat, diseases and see if we can help people at an individual level even. And so I think epigenetics is kind of a really good target for potential therapies because, again, it's modifiable, right? We can change our DNA methylation. We can change the expression of genes through microRNAs or integomeres or things like that. And so I think there's, this sort of ability for us to kind of, pursue that type of research as well. And how can we, utilize this mechanism to really be able to treat individuals?
Need to say that just follow up. So we know that for brain, many neurological disorder and psychiatric disorders, the histone modification, like drug against histone modification, has been used clinically for years, like SAHA. There's at least our reagent has been used. But the problem, they are not specific enough, often target like in higher genome regions. And the other thing is that how, for example, we are tracing to the genome editing specific loci, but we are linking the link between specific loci to their function and to the disease. So I think the connection in between is still missing. And the approach at this moment, we do have drug, but the drug is targeting entire genome broadly, and that causing a lot of side effects. You may affect in the memory. For example, you block DNA methylation, you lose memory, and you change that and using some drug, then PTSD or these can be at least change their situation. But how specific that is or not affecting others, that is a big question now need to address. So I think we are not just looking for drug, it's that looking for specific drug. But that specific drug often need the specific understanding on the specific loci, specific mutation, and for example, what you're talking about, that mutation, a piece of histone tails change, and how that really work. And the molecule, what you are doing now, for example, you need to be specific targeting that area, that region, and then change that. So I think specific things can be done, and that specific drug will come.
It's very complicated.
Yeah.
Well, I was going to say, I think you hit on like basically my, whenever I talk to students or, the public, kind of one of my first slides, what is it that I do? And that's the thing. It's like discovery and identifying what are those things that are associated with disease, characterizing or function, like, okay, let's change things around and see what does it actually do.
Mm-hmm.
And then there's the therapy. Can we then utilize all of that to, for treatment? So I think, you're right. There's these sort of three areas and you've got to hit all of them.
Would you be nervous at going in and making a genetic edits given the level of complexity and the proportion of what's known and unknown about this complex web of causation between all these different players in controlling the genes?
That's definitely an area of concern. I think epigenetic editing is a way to alleviate some of that concern, but definitely not all of it. And I think that's why when we look at these sorts of editing techniques, we probably still have a long way to go before we feel comfortable using them, as a day-to-day therapy.
But it depends on the cause, right? For example, I will see today's talk on Alexi, for example, using small molecule to target the protein specific interaction domain. That could be really specific. Then that side effect will be minimum, I hope. Lucky enough, right? But epigenomic editing is like genetic editing. So whether you go to right target, whether you go to right cell type. So a lot of issues still there, but that needs
the safety, efficacy, like that's all still
and then whether you go to all cell type, you target specific cell type, you target specific stage. So there's a lot of question behind it. And
the sickle cell has been approved. The FDA has approved CRISPR therapy for sickle cell and thalassemia.
Yes.
This has been last year, last 12 months. This is out there now.
Yeah.
So we're getting there.
Yeah.
I was going to say, I wonder, if there are ways to partner with companies who all they do day and night is think about these therapies and test them, safety and efficacy, or FDA databases, right? Are there smart things we can do molecularly to try and figure out which therapies might be safe and effective for specific changes? I don't know. There might be some out there.
I would echo real quick, a big problem with chromatin is that there is an enormous amount of redundancy baked into that system. Your typical, when you think about signaling cascades, you think about the biology, you think about this protein causes this effect, causes something downstream, and then it's cancer. The problem with epigenetics is that it's not a 1, 2, 3 ladder. It is like a neural network where we have 7 layers that are all connected in between. And what's going on inside these connections, we have no idea. But everything regulates everything. And if you perturb one modification, you are in all likelihood going to perturb 17 other modifications.
And whether that's a
predict.
and whether that's a good thing or a bad thing, maybe all of them do need to be perturbed to have an effect, or maybe not.
Do you think
methylase inhibitors, the HDAC inhibitors that have been in clinic for a very long time, we have no idea what they actually do.
But it's useful. It's showing clinical effect.
Yeah.
But we don't know how bad and a side effect.
And I guess the other factor in that is the timing, right? Is there a certain critical window in that developmental time period where the therapy will be most effective or effective, right? And when this is being established.
Or are you going to just really be fighting the symptoms once the disease has developed too?
We just mentioned X chromosome a little bit ago. There is a very famous developmental disorder that originates from a mutation on the X chromosome. It only affects females because males are dead when they have that mutation. But females have a very severe developmental disability. It's called Rett syndrome. It's a mutation in MECP2. MECP2 is a methylcytosine binding protein. And there was a fascinating paper from Adrian Bird just a few years ago where they had a mouse, an adult mouse with MECP2 deficiency, has massive disorders, can't walk, can't interact, has this huge pose defects. They re-express MECP2 in an adult mouse. The mouse would have been dead in a week. They re-express that protein and the mouse start being a normal mouse. And that suggests that, at least for some developmental disorders, it's not about some early developmental window. Like, not all is lost. You can fix things even in adults. That paper was just so inspirational and so much fun to read.
And I think it's so dependent upon the disorder and,
you're absolutely right.
and all of that.
Similar thing done for Fragile X. So, but it depends on the cause, and then the strategy will be different, right?
Yeah.
So some of them cause is really specific. We know exactly what's going on, and your target is more clear. But for some of them, it's connected to so many things, and some of them happen in the early development events. Then it will change, like what you mentioned about the cell type, cell function. Then or not later on, you can restore the function or not. For example, when the kids have autism, how you know that better know it earlier and get intervene at early time point, then you will have better outcome.
But then you also have, I know, Paul, I don't know if you want to comment, we have, like diseases like Alzheimer's disease, which, affect people later in life, but probably have risk early on. And I mean, when is the best time to kind of try and think about treatment for that potentially?
Right. I think one thing is trying to, because people don't actually know, unless it's familial Alzheimer's disease, it's kind of hard to determine, should you do it. Just like they're trying to look for biomarkers to try to predict when mild cognitive impairment will start to set in, and whether you should try and hopefully start a treatment to lessen the disease effects, or even just treat symptoms before that sets in. Or like currently right now, they're trying to do that with more familial Alzheimer's disease, because you can generally get like a nice age range of when the disease will start to set in. So I think that would be a pretty interesting question to answer.
Because it's neuroscience, I can't not think about some neuron-specific stuff. And maybe it's not as neuron-specific as I think, because I'm always thinking things are unique to the nervous system. Turns out not to be. But some of these things we've been talking about are relatively long-lasting. Some of them even can cross generations to some degree. We haven't talked about that. But some of them must be very brief, like immediate early gene expression sets off a cascade of changes in the neuron. The immediate early gene is gone. And then that will happen again tomorrow or the next day after that. And these things happen over and over again, and they are not they're not slow or necessarily long-lasting. So does that make it hard to study when there's something that's kind of short cascade or relatively brief things? It maybe leaves a trace when it's all finished.
I think it's kind of hard, because you do have to think about like the timing of things, right? A lot of the studies, like Chris, I've done a lot of studies, epidemiological type studies, but these cohort-based studies where, we'll go out, we'll collect blood from individuals and we'll look at their methylation levels and like, okay, is it associated with this disease? I've done a lot of work with diabetes and obesity, for example, and so the very first thing is, okay, well, you have to be fasting when we collect your blood, right? Because if you're not, that's going to change glucose levels to start, but it's also going to alter your DNA methylation levels. Now, that's in blood samples. Like in the brain, you're potentially going to have these less immediate effects that you see, but timing is still an issue. And, one of the downfalls of looking at postmortem brain tissue, for example, is, when you're trying to study like expression of genes or epigenetic mechanisms, there's always this question of, did this arise before death? Did this arise before the disease? Did it come after death? Is it, where is that change happening? Or more importantly, when is that change happening? If someone dies from a drug overdose, for example, is it due to the drug overdose or is it just due to death? Is it due to having, a psychiatric disease which led to the drug overdose? And there's so many kind of questions. that are hard to answer. And, one of the best things we can try to do is have very large cohorts, very large groups of people that you analyze, and you hope that you wash out some of those like variables so that you get a little bit more information about what you're really trying to look at.
You have these methods with incredible spatial resolution on the genome, but they're just temporally terrible. You can't do that again and again and again every 15 minutes.
Not with the brain.
So I guess, I had a question for y'all. Like, do you think gene expression, it being basically like how you said, just a snapshot of the cells, even if it's in vitro or in vivo, like a mouse really quick, it's just that snapshot in time. Do you think like epigenomics, like methylation or histone, changes would be a better marker of a disease compared to gene expression?
So when you say better marker, you're probably looking for a combination of how good this is versus how cheap this is.
Right.
How easy it is as well.
So
it can be analyzed pretty
it has fantastic drugs for Hep C, but they're incredibly expensive, and so not everybody gets them, for example, right? So RNA analysis is very easy, very high throughput, and it's been perfected to this point. Things like ChIP-seq require an enormous amount of material, enormous technical expertise. And so even if they are potentially a better predictor, there's no way they can be ported into clinic outside very specific use cases. I would, though, very quickly go back to these immediate genes. Everybody has looked at ARC and FOS, and I don't work in neurons, but I've seen the word ARC and FOS like 100 times. And it's one thing to measure the expression in response to stimulus, and it obviously goes up real quick and goes down real quick. But there's something happening at these genes early on, before the stimulus is there, that primes them for this activity. And things like paused polymerase, for example, it's there. There is already a machinery assembled at the promoter of this gene. It's just waiting for the kinase to come in and quickly activate it. So sometimes you can predict the response of a gene before the stimulus happens. You can identify that is going to be the responder. You can do that through epigenetics.
That's another kind of epigenetic control. It gives you temporal control over the gene expression.
And I think, to add, I think my sense, at least for methylation, is that there may be some patterns that are relatively stable, right, over time, and there may be some that are much more dynamic, temporally dynamic. And there's at least some evidence for that happening from like Andrea Baccarelli's lab, where they kind of gave an exposure really quickly in the morning and measured kind of every hour, right, at the methylation changes, and really showed, there's some of them were pretty stable and others were more dynamic. So I think that might be part of it.
And about your hydroxyl methylation side, right? They are like intermediate acting.
Yes, and then I think that's why, hydroxymethylation was kind of largely ignored for so long, because everyone sort of thinks, it's just an intermediate, it's going to go away, it's not relevant, it's not important. And then, through these neurodevelopmental studies, it's like, well, hang on, it's not really just going away, and we can't get this process unless that happens. So maybe it is, more important. And then again, I think when you start to look at neurons potentially, that you may see this sort of accumulation, but there's no further kind of oxidation and no degradation. And so that they stay there, and potentially, either serve as markers or, potentially kind of a mechanism for, changes within the cell.
What would you say is the, prospects for rapid progress? I mean, what would be the strategy? So it seemed, for me, watching what you're doing, it seems sort of focused on trying to identify the genes that are being controlled, and then what they do and what controls them. There are 29,000 methylation sites. So it's kind of a big job if what you're going to do is enumerate every single one of those and what they do. But just as an observer watching seminars and talks, I get the feeling that that's what's happening. Is that Is that going to work? Is that what we want to do? Is just to build a catalog of this stuff? Is that a good starting place?
We do that, right? For example, now you're talking about cell type. We do a lot of single cell analysis these days. Like you see that many cell Nature Science paper about sequencing brain cells. Like Allen Institute, they sequence thousands, and even million cells. That is a book, right? But now for us, individual labs is how to prioritize. Like we have, you see that we have tons of data. Even my own lab, I can analyze all my data and then do all. For example, you pull up today, say 5-6 transcription factor, and for me in the talk there, right, I show you I work on ER1, just one gene. But in the list table, I show you there are multiple of them. But I have to pick one to work on. So I think that it is a book, but I think everybody reading the book, everybody pick what they want to do, right? So, and then for you.
Yeah, you have to prioritize things and you have to have a reason for why you're prioritizing
and then,
that particular gene, for example.
Yeah. And particular side you want to edit, why you do that, right? So I think a lot of, for ourselves, is a decision maker is that how you decide what to do and what kind of strategy to use to address that question and how that links to disease and how the whole puzzle you can come together. You wish you would do a good job on that, right?
As long as we're doing them one at a time, we're really just hoping to hit the mother lode. I'm going to find the one that has Alzheimer's disease or something like that, and not the one that just is a helper to 25 others or something. Is there a way to maximize our chance of success?
Study process and not the protein. Study a biological question, and don't study an amino acid or a mutation. Because at some point you will exhaust all the biology that's potentially relevant to that one mutation. But if you're studying, let's say, transcription, I'm sure there will be a question about transcription 100 years from now.
I think I agree with that. I think even most of the therapies we haven't given a lot of redundancy, right, is that, it's probably not going to be just one or even a handful, but you have to think about how maybe the sets of individual sites you're finding might have a common pathway or downstream biologic process, right, that could be targeted.
So a lot of the time we'll do like these, WGCNA analyses to see what's kind of the correlation between all of these changes that are going on. Do they form a specific pathway? These genes that are all getting switched on or off at the same time, what are they actually doing? And then can you, maybe you don't just edit like one gene, but maybe you find what's the terminal part in that pathway that we can change, and thereby, maybe not worry so much about these, additional off-target effects. I think.
Go ahead.
Okay, so
I'm going to shift gears a little bit.
Detour here, and I would say that we should take a page from Adam Smith, and we should do the separate division of labor, right? There are some people in some labs that are really great at one thing, and maybe don't have expertise in another thing. And where it would take me 10 years to learn mouse models and whole genome analyses and neuroscience and retro elements and patient cohorts, science is incredibly collaborative. And most people love to collaborate and lend their expertise. And I think it's evident in the publication record these days because 50 years ago, papers had one author, two authors, three authors. There's one lab that comes with a Nature paper, boom, they discovered something big and important. Nowadays, you look at every Nature paper, it has 30 authors. And it has 30 authors because more often than not, some author made one panel, another author made another panel. The big trick is to put it all together and ride this big project. But I think we are in a stage when, if done right, division of labor really helps with how we run this.
Just follow on that, and then especially on the leadership, in university or college level, and even broader aspect, like how to maximize the output. A lot of time depend on how we distribute the resource. And nowadays, follow on that, some of our labs, they outsourcing their work to expert companies. So we probably eventually, I can see that the migration of the field eventually will have outstanding company or outstanding facility. They're able to offer outstanding work. And then it becomes shared facility, shared lab resource. And then we each on the lab, we don't need to keep a technician to learn each individual technique. It's just so for my past 10 years, I would say that just so painful for a lab to keep couple technician know everything. That's almost impossible. It's not going to be sufficient. Eventually, it will be replaced by collaboration, by shared facility, by shared resource. And how to develop these at a level, I believe that foreseeing the future becomes super important.
But I think from some level too, it's important that even though a technician or especially a PI doesn't have to know how to do every single thing, it's so important for them to understand the process and the biology behind each of those different experiments. You collaborate with people all the time. I have, I'm a molecular person, so I collaborate with, bioinformatics and statisticians.
Yes.
But I always want to collaborate with those bioinformaticians or statisticians who have done some wet lab research and have some understanding, because then, we can kind of have a conversation back and forth about, okay, well, these are the controls. And it's like, well, why are they the controls? And then you can explain it and they have an understanding. And then when they say, hey, these are the results. And I'm like, but hang on a minute, you didn't account for this variable. It's like, oh, okay, I need to do that, And so you can kind of have this back and forth because you have a little bit of an understanding of all of the processes.
Exactly. I totally agree. Totally agree. Yeah.
I love it. You ended up giving us two hopes. One is that the dimensionality of the problem will be reduced because all of these 29,000 sites are not independent of each other, but they're grouped together in groups that work together. And the other one is the dimensionality of the scientists will explode by having people go off in different directions. They combine them. Yeah. So that's a great way for us to end. So thank you, everybody. And this has been Neuroscientists Talk Shop.
Thank you.
Thank you.
Well, thanks for having us.